Yes, so thank you, Andreas. Thank you, Georg, for inviting me to this fantastic meeting. These were really amazing talks and showed us the possibilities of CAR T cells, but I also like to point out that the amazing clinical data we've seen in autoimmune diseases, in hepatitis B, also in liver transplant, were all clinical trials that were conducted in other countries and not in Germany. So a little bit complementary to my prior speaker, I like to also take the opportunity to point out that there are some challenges we are addressing in Germany to conduct clinical trials, IIT trials. So these are my disclosures. And I like to start, and I think the previous talk was a well introduction to this topic, address a little bit the topic of the principal investigators, the clinician and medical scientists who actually drive these clinical trials and have to have a lot of resilience to conduct this and perseverance if they want to follow through. And therefore, I'll like to start with just showing you the regional variances in the number of clinical trials that are conducted in the CAR T cell field, and highlight that there is some kind of career stagnation if you do clinical trials, at least in Germany, as your main focus to advance. I also like to introduce one of the, I think, really fascinating clinical trials that we are conducting, and that was already introduced by Fabian Müller as the first trial, and highlight a few words to also how this translates possibly into solid tumors. So come back to malignancies. And then at the end, maybe also insert some thoughts or discussion that I think that we need to move into translational hubs, a consortium, to advance this really exciting and expanding platform. So if you look at CAR T cell trials worldwide, looking at clinical government trials, and you look at 2019, you see the main players were US and China, and if you look now at 24, you see the increasing number of clinical trials in the CAR T cell field. And if you look a little bit further in phase 1, 2, 3, you can see okay, ich muss noch ein bisschen Geduld haben. You can see that China is leading the phase 1 clinical trials, followed by the US and then all the other countries. And if you look a little bit further and look at industry versus non-industry funded clinical trials, and you look at the top 5 countries, you can see that in the US as well as in China, sort of funding industry versus others is almost half-half. And then if you look at Germany, most of our clinical trials are funded by industry and not by other sources. Although I think in principle we are privileged in Germany with our funding agencies and DFG funding about 25%, which clearly is really generous. If you look at the investment in research and the increase over the past years, you can see that Germany is down here, whereas US and China at least have increased over the past years. And I suggest that clinical trial funding has to be increased in Germany. Also, if you look at the time frame for approval of a clinical trial, so one has to envision once you apply for funding, for example, for BMBF, which is sort of the largest funding agency in Germany, you have to have already a very solid package of preclinical data. It's very competitive. So then 2 years later, and this is an example from our own trial with adaptor CAR T cell, which I will introduce to you in a little while, but it took us 2 years from putting our proposal in until finally getting the money, and then it takes another 6 to 8 months to actually have the funding at your university available. So this is a very slow process if you think of the competitive and fast advancing clinical trial landscape that I think needs to be changed. So if you think about the persons who are actually driving these clinical trials, so our clinician scientists, I'd just like to highlight that this is challenging as well. And I think everywhere worldwide, we have a vanishing of the clinician scientist species. And we need to be aware and change things so that we keep academic medicine attractive also for medical doctors. So I think there are problems in the structure of evaluation in academic medicine. And I also think we have clearly a challenge in the infrastructure we are providing, and I think that was also nicely shown by Michael Schmidt in the previous talk. So just briefly, without going into the details, but I think in the past 5 to 10 years, at least in Germany, I think everywhere, clinician scientist programs have been introduced, which are now sort of established and not based on third-party funding anymore, where clinician scientists get a 3-year program with 18 months of protected research time, and this can be done within the sort of residency fellowship, but actually what is really, really popular and where we get the most applications is in the advanced clinician scientist program. So later on, when you already have your own group and you're trying to, for example, translate your preclinical finding into a clinical trial. Again, there's an 18 months protected research time, and I think all universities now have adapted what is sort of wanted by, I don't know, Generation Z or a little bit earlier, flexicurity, so maximal flexibility in how you conduct your research. Either, you do it 50-50% over 3 years, or you start 6 months only research followed by 50-50. So we provide everything also in different percentages if you have, taking care of kids or elderly or something, and a lot of security within this program. And all of this is aiming to support clinical research, translational research, and advanced academic careers, and give them a long-term perspective and attract them to academic medicine. But the problem is, and we're talking about clinical trials, is that the evaluation of clinician scientists is based on publications. So if you look, this is really strange for our US colleagues or from other countries, we still have to have a habilitation, so some kind of research qualification that is based on the number of papers, and it's in most of the university clinics sort of the prerequisite to become an attending or section or chair of a department. And there the number of publications are counted, and at least half of them have to be first or last author. So in none of these evaluations, it's asked if you've conducted a clinical trial, have been a PI or something. So this is under-evaluated. And just as an example that I'm not making this up, if you look at the ERC grant application, sort of the most prestigious sort of grant you at least can acquire in Europe, the only thing people are asking for are the excellence of your prior publications. So no word on clinical trials. So just last examples, if you apply for section or chair heads, everything that has been asked in all these sort of tables and so on as third-party funding and publications, and it's less about clinical trial or if you've done an IIT. So the question is, why don't you just publish your clinical trial data? I mean, if that's the value or the currency we are applying. The problem is that most of us are lost in translation. I really recommend this movie. And just an example from our experience, and that goes back to 2013, when we conducted an RNA transfected dendritic cell trials in acute myeloid leukemia. And we were really naive. We were doing this for the first time, and I don't want to put any blame on the Paul-Ehrlich Institute, but it took us quite a while, and you have to think of it, here we started for applying for funding, so we had already a preset of experiments where we wanted to head, and then we got our funding, and until we got it to PI and ethics, and then there was a problem. It was a small problem, but it took us another year to solve this problem. So until we finally got approval to do this clinical trial, it took quite some while, and it doesn't make you competitive, and it's boring once you start the clinical trial, right? And again, just to highlight that we probably are not the only ones who are struggling with this. If you look at BMBF and the number of clinical trials that have been funded, looking at 2005 to 2013, 81 clinical trials, and then 14 to 24, 80 clinical trials. So amazing number of clinical trials that are being funded. And if you then look at the publications that came out of it, just thinking also, about the clinician scientists who are giving their life to pursuing these clinical trials, It's represented here in gray, and you can see that I think the number of publications that came out of this is underwhelming and doesn't make it an incentive to pursue a career in clinical trials. So just to highlight that there is change happening currently, and there's this change and switch in the mindset. So at least the DFG, and they are determining sort of politics, research politics, they now just this year have entered that you can at least integrate clinical trial experience into your CV when you apply for funding. And I think, to my knowledge, the only Habil-Ordnung, again for the Germans, of the University of Tübingen, they have now entered, instead of just counting the publications, that you can, instead of a publication, also substitute with being a PI in a clinical trial, and that this is valued also in the context of this sort of qualification you need to move on. I think another problem, just one slide, I don't want to go into detail, is also that a lot of these requirements ask you to be first or last author, but I think looking at all the stuff we've seen in the past 2 days, it's clear that most of the things we're doing now is team science. So I think we also have to think how we value co-authorship, or if you have 4 first authors, 5 last authors. That's the reality of competitive science, clearly independent of clinical trials. So the second aspect, I think, that has to be addressed is that we need a better infrastructure. So a clinician scientist, or somebody who's doing translational work, is overwhelmed by really moving this through preclinical GMP and into the patient, particularly in the area of cell and gene therapy. And I'd like to highlight one positive example that is at least trying to evolve in Bavaria, where we have a consortium of 6 universities. And in the context of cell and gene therapy, we have assigned each university clinic sort of one topic, so that one university is providing the expertise for a specific topic that is needed for moving a cell gene therapy forward. And for example, we are sort of responsible of toxicity managing, providing SOPs to everybody that can be then applied by the other centers to move CAR T cells forward. We have regulatory affairs, GMP manufacturing, that's assigned in Erlangen. So trying to make consortia, decrease the hurdle for each university to move forward. And hopefully a success story will be our clinical trial in the context of AML, where we're going to use adapter CAR T cells for treatment of AML, and that's being done in conjunction with Erlangen. We're trying to get some gold dust also into Munich, because working with Erlangen seems to be the way to go. And this is also done in conjunction with Miltenyi. So in this trial, we're going to treat patients with relapsed refractory AML, which have a really dismal prognosis, but also patients with MRD positivity, which also have a really terrible prognosis with adapter CAR T cells. So here we have the advantage of generating a generic CAR T that is recognizing a linker label epitope, which is connected to a Fab molecule, and thereby we have the advantage with this platform to have multiple and sequential targeting. This adapter is given as a continuous infusion, so we stop the infusion, we sort of have an on-off switch of our CAR T cells. This also allows to have a resting phase, and has been clearly shown as one of the resistance mechanisms evolving T cell exhaustion. And we can work with different adapter molecules, thereby counteract antigen escape variants. And clearly, this platform possibly allows to further engineer our CAR T cells, but also move it to other disease entities. The disadvantages are that depending on the target antigen, there might be antigen sink of our adapter molecule, as well as the disadvantage of continuous infusion, which in the case of AML patients is less of a problem. They're mainly pretty sick and sort of like to stay in the hospital, but for other disease entities, that might be a challenge. We could show that these adaptor molecules are highly efficacious against primary AML settings. And also, and that was shown already by Fabian Müller, we are not the only ones working on the adaptor approach. So in a trial that is already running, phase 1, where they are also using an adaptor CAR T-cell platform, also in the context of AML, at least they had 2 patients where they could show that there was complete reversal of immune-related adverse events upon stopping of the adaptor molecules, so supporting safety of this approach. At least preclinically, as said, we could show that if you use T cells that have been exposed and then get a resting phase, have improved efficacy. That has been shown with bispecifics, but also CAR T cells. And you can also alternatively use a small molecule like dasatinib. And what we also could show, at least preclinically, that you use multiple adapters, you can increase efficacy and counteract immune escape. And at least in a mouse model, we could show that a conventional CAR T cell is similar in activity to our adaptor CD33-directed CAR T cells. So if you look at our clinical trial protocol, you already see that this is challenging. So we are going to integrate, we have to combine the adapter molecule and dose up of the adapter molecule. We have a step-up dosing of the CAR T cells. And in addition, we also want to step up within our patient based on bone marrow assessment to increase efficacy. So there will be step-up dosing within the clinical trial. So a complicated trial result where clearly a clinician scientist needs help. I think this adapter platform, but also in principle, and we've heard so many really exciting talks during this meeting, allows for multiple further engineering of multiple CAR T cells, to increase specificity depending on the construct. If we use split CAR T cells, we can increase specificity. With dual targeting, we can increase efficacy. There are CAR T cells with safety switch or double knockout receptor ones. We have cytokine-secreting CAR T cells to increase efficacy. And I think this is really a interesting area where we can sort of use CAR T cells as a vehicle to translate cytokines or whatever. They are also bispecific secreting CAR T cells to the site of the tumor and thereby possibly also modulate the tumor microenvironment. And there are a lot of clinical trials running in this context. I think one of the most advanced was presented here, again, in the context of CD19. So here, IL-18 secreting CD19-directed CAR T cells in the context of lymphoma. And first of all, they showed safety. And second of all, these were all patients that had been previously treated with CD19-directed CAR T cells. And if you consider that, they had very interesting activity. And to my knowledge, there is a clinical trial starting in Germany with IL-18 secreting CAR T cells in pediatrics as patients with glioblastoma targeting GD2. So things are also moving in Germany. Fabian Müller already presented some of the data of CAR T cells in the solid tumor, where we clearly have the challenge of identifying suitable target antigens. And I just want to highlight one other aspect, how things are moving in the solid tumor field, and these are regional delivery of CAR T cells. And you can see that different target antigens in different disease entities are being followed with interesting results. What's also really interesting is that the local application leads to systemic detection of CAR T cells, and here you can see regional anti-methylstalin CAR T cells that are sort of, here you see the injection sites, and at least it shows feasibility. It shows systemic circulation also of these CAR T cells. And what I think is really interesting in this context, but also in principle, combinatorial approach in this context with inhibitory checkpoint molecules. And with this slide, I sort of want to sum up the activities of the CAR T cells. I think within this meeting, we've heard almost all, for all these disease and entities, examples of successful moving forward in clinics. We started with data on the malignant disease. We heard a lot about the excitement in autoimmune diseases. We heard talks about fibrosis, senescence, and also infectious diseases. And I think it's clear CAR T cells are not only here to stay, but also to expand. And that's why I think it's worth to invest in translational hubs. I showed you one example of Bavaria, but I think in principle it's too small. We have to think on national and even international level. And I think what is needed is that people who have great ideas, great findings in the lab get support to move it into a phase 1 trial. And I think we need centralized structures. It's not really fitting, so maybe it's a perfect example for Germany. But where things are moving together that help each sort of a scientist who's working that direction to move this into clinics. And clearly, I think one of the key things is that we need clinical trial centers with expertise for cell and gene therapy and the caveats and the hurdles that have to be overcome. And clearly, I think where academia belongs is developing CAR T cells and novel entities. I think it's not our task to give, 10,000 people CD19 CAR T cells and lymphoma. Actually, I think, our task is to move things forward, and that's what we need. And that's a perfect example from Erlangen, which would never been possible if they wouldn't have had their own production site. So I think these are key issues because, most of the patients, at least in malignancies, will relapse after CAR T cells. So we have to improve to increase the number of patients that will benefit. And I also want to thank our patients that are so courageous in entering all these clinical trials. And for those who are not doing CAR T cells, so we talked about it yesterday that there is neurotoxicity or can happen, and particularly in lymphoma patients. And one of the sort of assessment, if they develop a neurotoxicity, like a 10 questionnaire in one sentence. So these patients for 10 days or longer, 3 times a day, have to write the same sentence. So I'm collecting these sentences of these patients, and this is just to acknowledge to the brave patients that are participating in our clinical trials. And with this, I'd like to finish. Thank our collaborators. Thank Daniel Nixdorf, who did the data I showed you in the context of AML, and thank you for listening. Thank you.